Hey, this is Mr. Buss, and in this video, I'm going to demonstrate the in-class activity we did with pop beads, and we modeled the DNA structure and the process of DNA replication. So these little pop beads, we got a lot of different colors of them, so I just arbitrarily chose yellow to represent a guanine nitrogenous base, so not the whole nucleotide, but the base. And then I believe used, so cytosine is purple, so guanine and cytosine are gonna be paired up. And then adenine was blue and thymine is orange, so thymine and adenine are gonna pair up. So those two colors are gonna pair up. And then I used white pop beads for the sugar. I just thought, sugar white would work. And then phosphate were the pink beads. So we're going to use the beads, and then we're going to use these little plastic connectors available as well as a way to show the hydrogen bonds. Okay, and so we're going to go through this in a couple parts. First part is just going to be building up a DNA structure. And then the second part is going to be going through the process of replication and talking about some of the enzymes. So one thing I wanted to make sure that this model, this activity showed well is the idea of directionality. And so the pop beads have a directionality to them, right? And so like the sugar molecule here has a direction kind of pointing towards the button there. And so like we know that in class we talked about how the ribose sugar for deoxyribose sugar has a 1, 2, 3, 4, 5 carbon. Well, we're going to say that the 5' end, the 5 carbon is going to be on this end, and then we're going to say that the 3' end is over here. So we are going to assign a directionality to this. So the 3' end is going to be that end, and the 5' end is going to be this end. And so when we put these together on the sides of the DNA molecule, I ask students to make sure that they always put the pop beads pointing in the same direction for one of the sides. So if I'm going to build the sugar phosphate backbone of one of the sides, I'm going to make sure that the pop beads are all going in the same direction for one of the sides. And so, for instance, something like that, where they're all pointing in this direction. And then as I build the other side, so I'll just kind of just show this quickly, I'm going to do the same thing, put them all in the same direction. Let's do a couple more here. But then when I put the strands next to each other, I have to make sure that that they don't run in the same direction, that they run in opposite directions of each other. So anti-parallel, right? This strand is going 5 to 3, 5 to 3, 5 to 3, so that's in this direction, and this one's going 5 to 3 in this direction. And so rather than have you watch me build the whole thing, I just brought a finished product here. So this is the idea then, is that we're gonna have this strand running in this direction, this strand running in this direction, alternating sugars and phosphates. The phosphates are the spacers basically between the sugars, and then the hydrogen bonds are shown with those connector molecules in between. And just again, just to show like what is one nucleotide, right? So this would be one nucleotide, right? One sugar, one phosphate, and one nitrogenous base. This one would be thymine, and this would be adenine, for instance. So I'll put those back together. But that is a pretty good model. And then the nice thing about the pop beads too is you can kind of show the twisted ladder or double helix nature of it by just simply taking the ladder and twisting it to show more accurately a three-dimensional representation of DNA. So the next step I had students look at was the replication process. So what I had them do is I had them break apart most of the bonds, the hydrogen bonds. So the covalent bonds between the sugars and phosphates aren't breaking. It's the hydrogen bonds between the A's and T's and the G's and C's. And so helicase enzyme is what does that. And we're not showing the replication bubble, the origin of replication. A replication bubble would open up. Single-stranded binding proteins would attach to keep the strands from moving together, and then a helicase enzyme would go in each direction, and it would start to break apart those hydrogen bonds. So we're kind of at that phase. So I had students breaking apart 7. Okay, of those, and then I'll just put those hydrogen bonds, right, those are the hydrogen bonds. And so there's the helicase enzyme, and then this was the second picture we took, is showing how the helicase enzyme basically unzips the DNA molecule at the hydrogen bonds, okay? And then the third picture we took is like a further continuation of the process of replication, so going beyond just the unzipping of the strands, then the rebuilding of the strands. And so then we have DNA polymerase enzymes, and those polymerase enzymes connect to a primer. So the primer isn't shown, so the primase enzyme puts a primer on there that's not shown. And then the DNA polymerase is going to continue to add nucleotides at the primer region. And so what I had students do is then build the double-stranded new sides. So I'll use these pieces that I built here, and I'll put the correct bases in between them. So, so the nature of anti-parallel nature we need to keep intact. So if this strand is moving in this direction, then we're going to make sure that this strand is moving in this direction, and then we can put in the correct nucleotides and build those, and making sure that we're connecting the nucleotide bases to the sugars. So we did oranges and blues, and yellows and purples, and this doesn't take too long to put together here, and then I'll be able to talk about the finished product. Okay, so this strand is done, and then this strand is going to be built in this direction. Now here's the replication fork, here's the helicase enzyme. And so this polymerase enzyme is going to hop on at a primer, and it's going to move in this direction away from the replication fork. What that means is that this is going to be the lagging strand, okay? There's going to be a continuously growing gap as this moves in this direction. There's going to be a gap of single-stranded DNA here, where another DNA polymerase enzyme is going to have to attach to another primer. So the lagging strand makes fragments, Okazaki fragments they're called, and the direction away from the helicase replication fork. This is going to be the leading strand then. And so this one is going to, since this is here, this is going to be like that. And then I'm going to go ahead and just quickly put these together. So I'll need yellow to the purple here, which would be yellow is guanine and purple is cytosine. Put those together. And then blue is thymine. And I'm sorry, blue is adenine, so I'll need adenine to the thymine here. And let's just do one more. Another blue. Adenine to the thymine. All right, excellent. Okay, so this is going to be the leading strand. So the DNA polymerase, once it hops onto a primer on this side, since this is going 5' to 3' in this direction, this polymerase is going to be moving towards the replication fork, adding new nucleotides to the 3' end, just like this adds new nucleotides to the 3' end. Actually, that would be more like this, of the sugar here. So this is going to be building, and this is going to be building. Okay, and so this is the leading strand, this is the lagging strand, and that's DNA replication at its kind of base level. We kind of are skipping past some elements like topoisomerase, which corrects for the twisting nature of this. I can show that pretty quickly, actually, though. Let me get I have a larger DNA molecule here that I can use to show. So topoisomerase, I can just show. Here's the DNA molecule, if I kind of show it twisted here. Okay. As the DNA molecule becomes replicated and broken apart, the helicase enzyme tends to supercoil the DNA, and it tends to form a really tightly wrapped region of DNA. And topoisomerase actually breaks apart a covalently bonded sugar and phosphate in order to allow it to unravel. So this actually model can show that pretty well. I'm not sure if I'm showing it well in the video or not. And then it'll reconnect those broken backbone pieces. But it kind of ignores some aspects of replication. So I hope this was a helpful video for those students that missed the DNA pop beads activity in class. Take care.